Now let's concentrate about Ras-Raf-Map kinase pathway. First of all, activation of receptor tyrosine kinase. It may triggers activated RTK, may trigger some pathways. Activation of EGF receptor activates RAS dependent pathway as well as RAS independent pathway. Activation of EGF receptor can activate RAS dependent pathway as well as RAS independent pathway. This RAS independent pathway involves second messenger. RAS activation triggered by activated receptor tyrosine kinase. These receptor tyrosine kinases, they are short-lived. Activate a downstream serine-threonine phosphorylation cascade. Includes a MAP kinase. Full form of this MAP kinase is mitogen activated protein kinase. Induces a kinase cascade that culmination in activation of MAP kinase. This serine-threonine kinase can translocate into nucleus, phosphorylates many different proteins including transcription factor TA. This regulates expression of important cell cycle differentiation specific proteins. Summarize the whole. This is about Ras-Raf-Map kinase pathway. Lots of unknown unusual terms are used here. All of is initiated by an activated RTK, receptor tyrosine kinase. After being activation of RTK, it causes activation of EGF factor. EGF means epidermal growth factor, and this EGF factor activation, it now can activate two pathways. First one is RAS dependent pathway and second one is RAS independent pathway. And the second pathway means RAS independent pathway, it needs second messenger. It cannot work of its own. RAS is being activated, and after activation, this RAS activation is triggered by RTK, activated RTK. It is very short-lived. RAS is being attached with GTP now to being activated. Then what happens? This after activation of RAS, it activates a downstream serine-threonine phosphorylation cascade, and this serine-threonine phosphorylation cascade pathway, it includes, means it needed a MAP kinase. MAP kinase means it is an enzyme, okay, and its full form is mitogen activated protein kinase. So what they have done, what activated has been done, active a downstream serine-threonine phosphorylation cascade and induces a kinase cascade also, okay. Then activation of MAP kinase occurred, okay. MAP kinase, it is a serine-threonine kinase translocated into the nucleus and phosphorylate. After translocating inside the nucleus, it now phosphorylates many different proteins, transcription factors, okay, and these different proteins which are being phosphorylated by this MAP kinase, MAP serine-threonine kinase inside the nucleus, they can now regulate expression of important cell cycle and different specific proteins. Now, the activation of MAP kinase in two different cells can lead to similar or different cellular responses, as can activation in the same cell by stimulation of different RTKs, receptor tyrosine kinases. Many serine threonine kinases participate in this phosphorylation cascade, but 3 of them constitute the core module of the cascade, and the last of the 3 is called MAP kinase. Got it? Last of the 3 is called MAP kinase. What happens then? Many serine threonine kinase can participate in this phosphorylation process, but only 3 of them can constitute the core module, and that 3 serine threonine kinase which constitute the core module, it's known as MAP kinase. Is it very clear what is MAP kinase and how it's going to work? Now, downstream serine threonine phosphorylation cascade follow a pattern. Okay, now let's discuss about that pattern. Now, see the diagram. Activated Ras binds to RAP. This is plasma membrane, and in your exam copy you should draw this like this fashion. It's PLP layer of plasma membrane. GTP bound Ras, and this is activate Ras protein. First step of this pathway is attachment of activated Ras protein with RAP. That's why the name Ras-Raf-Map. This is inside the cytosol. Now come to the point, what is RAF? Full form of RAF is rapidly accelerated fibrosarcoma. Rapidly accelerated fibrosarcoma. And this gene, this RAF gene was first described as a retroviral oncogene. Retroviral oncogene. It means gene responsible for creating cancer inside our body. And what is this retroviral? This is a totally separate conception which will be discussed at the time of discussing cancer. VRAF by the murine sarcoma virus isolate. This was transduced by the murine sarcoma virus isolate. So this information is sufficient for this pathway. Okay. So now activated GTP is bound with Ras. Serine threonine phosphorylation occurs. It changes into MAC. This is mitogen activated protein kinase. Tyrosine threonine phosphorylation occurs. Tyrosine threonine phosphorylation occurs. MAP kinase is formed. MAP kinase. And we are all familiar with this MAP kinase. Again, serine threonine phosphorylation occurs. Changes in protein activity observed, or changes in gene expression occurs. So this is the complete Ras-Raf-Map kinase pathway. First of all, activated GTP now can bind with RAF, and RAF is rapidly accelerated fibrosarcoma, and it is a retroviral oncogene. MAP triple kinase. MAP kinase. Okay. Many serine threonine can participate in this phosphorylation process, but only 3 can made the core module. So these 3 are core module. RAF is nothing but a serine threonine kinase. It's nothing but a serine threonine kinase. So among many serine threonine kinase, only 3 can constitute the core module of RAF. Is it now very clear? Now see the next point. What about MAKE? MAKE is, constitution of MAKE is MAP kinase, two kinase. One is reduced because serine threonine phosphorylation occurs here, and one kinase is lost and RAF changes into MAKE, mitogen activated protein kinase. Here RAF directly phosphorylates MAKE. This is an unusual enzyme. This is an unusual enzyme. It has dual specificity. MAKE has dual specificity which can phosphorylate both threonine and tyrosine. Both threonine and tyrosine. Its target is the ERK. This MAP kinase, this one actually is known as ERK. Full form of ERK is extracellular signal related kinase. So first RAF, then MAKE, then ERK. Kinase, 3 times kinase. Here MAKE is MAP kinase and then ERK MAP kinase. Okay. MAKE can phosphorylate, activate MAP kinase. Phosphorylates and activate MAP kinase. MAKE finally phosphorylates and activate MAP kinase. MAP kinases are serine threonine kinase. These MAP kinases are serine threonine kinase. The MAP kinase is a serine threonine kinase, and this changes in protein activity or changes in gene expression. But between these two steps, there is lot of information. I am going to erase all this. Target. What is the ultimate target of this pathway? MAP kinase phosphorylates many different proteins, including nuclear transcription factor, and that mediated cellular response. The ultimate effect of MAP kinase pathway is ultimate effect of MAP kinase pathway. Change in the pattern of transcription. In the classic pathway, MAP kinase pathway, MAP kinase move into nucleus. Here at nucleus, MAP kinase phosphorylate target transcription factor. In case of alternative pathway, it phosphorylates MAP kinase, it phosphorylates cytoplasmic factor. In classic MAP kinase pathway, acts on nucleus and phosphorylate transcription factor, and alternative MAP kinase pathway, MAP kinase phosphorylates cytoplasmic factor. One pathway is for nucleus, another pathway is for cytoplasm. And this cytoplasmic factor, it may be also a transcription factor, and if it is transcription factor, it then moves to nucleus or a protein that regulates transcription factor. This is about all Ras-Raf-Map kinase pathway, its activation and its ultimate target. So it's all about Ras-Raf-Map kinase pathway, and this Ras-Raf-Map MAKE kinase pathway. This one is very much long, and as long as it is very much important pathway of signal transduction. Okay, hope you all enjoyed it. So stay connected.